This video will guide you through the first part of Chapter 5 of our Anatomy and Physiology text, the integumentary system, the skin. You can take a look at these introductory slides from the publisher, and we will jump right in and start talking about the integumentary system. First, we'll be talking about the skin, the epidermis, and the dermis. And then we'll be talking about some appendages, some like mini organs that form from the epithelium, the epidermis. So, first we'll talk about the epidermis, the keratinized stratified squamous epithelium. Then we'll talk about the dermis, which is connective tissue proper. And then we'll make a few mentions of the hypodermis, which is adipose tissue, which underlies the dermis. This cartoon is nice because it includes a representation of pretty much every different anatomical structure that we're going to encounter. Only downside of it is it's wildly out of proportion. But let me just take a look at a few things with you about this diagram. It's kind of a good summary tool. The epidermis, the keratinized stratified squamous epithelium, is in kind of orangish tan here on the surface. And you can see it forms like a wavy boundary with the papillary dermis that underlies it. We know that all epithelia have areolar connective tissue under them, and that's exactly what the papillary dermis is. Areolar connective tissue proper contains all the capillaries needed to feed that many-layered epithelium with oxygen and nutrients and so forth. The dermis not only has the papillary layer, areolar tissue, the vast majority of the thickness of the skin is dense, irregular connective tissue proper, which is the reticular dermis. That's the mechanical strength of the skin, whereas the epidermis represents the seal, kind of like the cell membrane of a cell, but the integrity of the cell comes from the cytoskeleton. The integrity of the skin comes from the reticular dermis. So let's take a little closer look. Not only are there lots of blood vessels, capillaries in the papillary dermis, there's some free nerve endings that give some of the sensory properties of the skin, such as pain receptors, temperature receptors, itch receptors. There's also some modified receptors in the epidermis itself, which we'll discover give us a light touch receptor function. There's another receptor, see way down here in the reticular dermis. It's a layered collagenous bulb and then a capsule around the dendrite or antenna of a neuron, and so it's called a Pacinian corpuscle. Or here your authors have called it a lamellar corpuscle for all the apparent layers of collagen on there. Pacinian, P-A-C-I-N-I-A-N is the correct name of it. The reticular dermis has a lot of redundant looping blood vessels, not capillaries so much, but larger vessels, and the function of that is in temperature regulation of the body. A lot of extra blood can reside in the skin and can flow through the skin, through the dermis, when we're trying to get rid of more heat from the body, say during exercise. We can make more blood coming from the deep, hotter regions of the body flow through the dermis, which is very near the surface, and it'll create a steeper gradient, temperature gradient for release of heat from the surface of the body. If we don't want to lose heat from the body, we can constrict all those vessels and pull all that blood flow back down within the hypodermis, this insulating layer of fat, so that the heat will remain trapped in the body. We'll see more about that later. A couple of glands that are accessory organs in the skin. Here's an eccrine sweat gland. Its secretory simple cuboidal epithelial cells are arranged kind of in tubular form, all wrapped up into like a ball of spaghetti, and then the secretion, the sweat water, travels out through a duct onto the surface of the body, making it an exocrine gland. The sebaceous glands are associated with the superficial most part of a hair follicle, and they secrete a lubricating oil, which will sebum, which will enter the hair follicle and then lubricate the hair shaft and soften and lubricate the skin surface. So let's take a look at the epidermis. What does it do for us? It provides a barrier between the inside of the body and the outside. It's a barrier against water loss. It contains some waterproof lipids that keep the moisture inside the body. Some evaporates out continually, but the vast majority of water stays in. It's a manageable rate of water loss. It protects our underlying tissues from abrasion. The skin, the epidermis, is quite resistant to scraping, rubbing types of injuries because of the keratinized nature of its stratified epithelium. It also is very impervious to bacteria and fungi, and of course viruses. They're not particularly good at getting through dead cells. They need live cells to take care of their life cycle for them. So the keratinized layers are the dead cell layers on the surface, as we'll see, and they're very resistant to degradation by bacteria, by microbes. Sweat contains antimicrobial proteins, so you're seeing that not only is the epidermis a physical barrier, it's a chemical barrier. Sweat contains antimicrobial proteins, slows down the growth of the bacteria that cover the entire surface of your body. Sebum itself is acidic in pH, and it's the lipids that comprise it are not very hospitable to bacteria, not particularly good for them to eat. So we have this acidic surface of our body which inhibits bacterial growth. Sometimes it's been historically called the acid mantle. I don't know if that term is still holding up very well today with physiologists, but that is a well-used term to describe that acid pH which suppresses microbe growth. Keratinocytes, the cells that make up the epidermis, secrete antimicrobial, antibacterial peptides called defensins. So there's more of that chemical barrier. The bacteria on the surface are there, and they are live, but man, it's rough go out there because of all these inhibitory events or conditions. Again, water loss, we've limited the water loss because of the lipid-saturated cornified layers or keratinized layers. UV injury, ultraviolet light, has enough energy to physically modify DNA molecules so that when they're replicated, they produce mutations. Incorrect copying happens. So melanin, a pigment in the epidermis, absorbs UV light so that it can't penetrate down to the deepest layers of cells, the basal layer where cells are dividing. All right. So sensation, we mentioned some of the sensory neurons. We'll see again, we'll take another look in a second at sensory neurons in the epidermis. Immune surveillance, the epidermis has Langerhans cells. They're a type of phagocyte, they're dendritic cells, they're called. It's a type of phagocyte that has receptors to recognize foreign agents such as bacteria, viruses, and so forth. And it will, once recognized, it will phagocytose the bacteria, engulf it, and kill it, and, interestingly, travel to the nearest site of lymphoid tissue, reticular connective tissue, and report to the lymphocytes of the immune system what it's found. Start up an immune response. And finally, vitamin D synthesis. Cholesterol is the starting material for making various steroid hormones and vitamins. And so in the epidermis, using energy from ultraviolet light, the first committed step of vitamin D synthesis occurs, and then that precursor travels in the blood to the kidneys where the final active vitamin D can be produced. But you need to have that first step taking place in the skin, or you can go to your nearest pharmacy and purchase some 2,000 unit gel caps of vitamin D. Highly recommended for those of us who don't spend too much time outdoors or who live in northerly climates where the sunshine isn't as intense. Okay, so let's look a little closer at the epidermis. What are the cells available there or found there? The keratinocytes are the name of the cells that produce the epidermis, the bulk of the epithelium. They're the building units. They are called that because they have this keratin cytoskeleton, which actually varies greatly in terms of the type of keratins that are present in the basal layer. And as we progress superficially, the final keratinized cells have a very tough kind of keratin in them, packed full. Keratinocytes are tightly connected from cytoskeleton to cytoskeleton. The keratin filament cytoskeletons are connected by desmosomes, and yet they are programmed to slough off at a prescribed rate from the surface of the body to be replaced from beneath. Melanocytes are the pigment-producing cells of the epidermis, and they lie in the basal layer of the epidermis, and they're just sprinkled along there, and they have long processes by which they transfer vesicles full of this brown pigment to the nearby keratinocytes. So the whole epithelium starts to get pigmented by this UV-absorbing melanin produced by melanocytes. Very great ratio, that's not the word I want, very great degree of a range of density of melanocytes in the population. So some people have essentially no melanocytes. They would be albino. Some people have very few melanocytes and they're very light-skinned. Some people have like orangish yellow melanocytes. They produce a kind of melanin, pheomelanin, that's a lighter color, but the vast majority of us have regular melanin-producing melanocytes, but the darker brown, rich brown your skin is, the more melanocytes you have. Dendritic cells, as I said, are those phagocytes that live in the epidermis. How they can move in and among the keratinocytes in spite of the desmosomes. I'm not sure how they exactly even can do that, but they do. And they are the first responders often to identify something foreign. If you make a nick in the epidermis, in the keratinized layers, those cells will be on the job identifying and reporting on foreign agents. Merkel cells are tactile cells, meaning sensory receptors that lie in the basal layer of the epidermis. And we'll take a look at a cartoon that shows what that looks like. So here's a list of the layers of the epidermis. It says 5 layers of skin. I'll need to edit that slide. 5 layers of the epidermis, which is probably a poor choice of words, layers, because when we are talking about the skin, we say the layers are the epidermis, the papillary dermis, and the reticular dermis, and then possibly you might mention the hypodermis. Those are the layers of the skin. But then when we look closely at the epidermis, it too has its own layers. So kind of a hassle with the word layer in there, but definitely I should not be calling this the layers of the skin, lest we should say something about the dermis. Okay, so anyway, let's look through those layers a little more closely and see what we're dealing with. The basal layer of the epidermis is where the dividing cells are. It contains stem cells, which are the origin of all the cells. Any tissue in your body that is constantly being replaced, blood, all the epithelia, and so forth, all of those compartments have stem cells, which are the origin of all those constantly reappearing new cells. So stem cells down there in that basal layer are interesting cells. They're unique among cells in your body. One of the characteristics they have that's unique is that they're immortal. All the other cells in your body can divide a finite number of times, and then they can no longer divide. They become senescent. Maybe 50 times they can divide, and they can no longer do it. There's like a built-in counter, but yet stem cells aren't like that. They're this close to being like a tumor cell. One of the comparisons is that they are immortal. They can divide indefinitely like a tumor cell can. They also produce asymmetric division. Most people that are familiar with mitosis or have studied mitosis have studied a process by which a cell can kind of replicate itself, and you wind up with 2 identical daughter cells that separate by cytokinesis after the chromosomes are replicated and separate. Well, stem cells can't do that because it wouldn't make any sense. We need a product cell that can populate the epidermis. So when stem cells divide, they produce one stem cell to recapitulate themselves, and they produce a daughter cell which is destined to go on and populate the epithelium, a transit amplifying cell. Those cells are in the basal layer, and they divide very, very rapidly, producing a large number of progeny, and then they eventually stop dividing. And they push cells superficially, and they themselves will move superficially to populate the outer layers of the epidermis. It takes about a month for a keratinocyte to go from the basal layer to the surface of the epidermis. Really nothing new here, probably, that we needed to jump back to this slide. 10 to 25% of the basal layer may be comprised of melanocytes, depending on how dark brown your skin is or how light white your skin is. The spinous layer is the next layer superficial to the basal layer, and as the cells push off from the basement membrane of the epidermis, they enter into a genetic program of differentiation or maturation, and they start to change. And the spinous layer is characterized by keratinocytes with super thick, heavy-duty desmosomes, cell-cell junctions that link the cells together, and they're so prominent that when you look at those cells in the light microscope, they look like they have spikes sticking out. And so this is sometimes called that's why it was called the spinous layer. The cells are sometimes called prickle cells because of how they looked to anatomists way back when in that regard. And then as the cells continue to differentiate and move superficially, we come to the granular layer. And the granular layer keratinocytes fill up with vesicles or granules. There's two types of granules that are in those cells. I'll show you in a micrograph in a second. You can really see those granules in the granular layer. Keratohyalin granules contain are just chock full of a mature kind of keratin that will form the tough outer covering of the epidermis. Also some other proteins, structural proteins that will form essentially what's called a cell envelope. Under the cell membrane will be cross-linked a whole bunch of proteins that form essentially almost like a little nylon bag, woven nylon sac in which all the keratins are going to reside, the tough intermediate filament cytoskeleton, and then we'll link those things together by desmosomes in the end. And finally, lamellar granules in the granular layer. Their lamellar means striped, and you can see how they look in the microscope. They contain lipids that will eventually waterproof the epithelium. So just before the cells die, they exocytose all the lipid in those lamellar granules, and they just all their organelles disintegrate, and the cell envelope forms under the cell membrane, and the keratin filament cytoskeleton forms, and then the cells are done. They're no longer alive, and they form layers of the keratinized or cornified layer. In thick skin, on the palms and soles, there's a unique few layers of cells that look transparent, which is called the stratum lucidum. It's not a physiologically particularly interesting fact. It just occurs that if you look in there, you'll see kind of a translucent region. But the outer layers that actually produce the barrier function of the epidermis, that's the keratinized or cornified layers, those are interesting. A great range in the number of layers, but layers of dead cells packed with tough keratin cytoskeleton filaments, attached together by desmosomes, saturated with waterproofing lipids, and piling up on the surface. And then eventually leaving the surface by a programmed process called desquamation. Right? Squamous cells, when they come off, that's desquamation. And there's proteases actually built into the keratinocytes, synthesized by the keratinocytes in the later stages, that slowly degrade the cell-cell junction, so that at the exact same rate that new cells are being produced and rising superficially, the surface cells fall right off the keratinized layers. So a wonderful architecture that's perfectly coordinated depending on where in the body it's located and how much abrasion it is subject to, and so forth. Here's a little cartoon of a desmosome, and it's like two little rafts of Velcro-like interlocking hooked proteins that attach at the exterior of two cells and connect them together, a dense plaque of proteins inside the cell membrane of each of the two cells forming the desmosome. And then here the orange fibers are the keratin filaments. So the keratin cytoskeletons of two neighboring cells are anchored together by desmosomes. The basal cells are actually anchored to the basement membrane and to the areolar connective tissue, the papillary dermis, by hemidesmosomes. They have essentially one side of the structure and then some long anchoring proteins that reach right down and connect onto proteins of the matrix of the areolar connective tissue in the basement membrane. There are actually some diseases, terrible diseases, in which the immune system aberrantly attacks proteins of the desmosome. Antibodies are formed so that it prevents the proper junctions from forming or being maintained. They're called bullous diseases because they lead to blistering. when there's not good adherence of the keratinocytes to each other, just the least amount of mechanical challenge to the epidermis causes the layers to split apart, and then water begins to infiltrate, forming blisters. So this is a picture of pemphigus vulgaris. It's called one of the more sort of penetrating disease states, autoimmune conditions of desmosome problems with the immune system. So the stratum corneum, in review, is layers of dead cells slathered with waterproof lipid that protect the deeper cells from the environment, namely chemicals, bacteria. We see bacteriological chemical protection against physical assault, ultraviolet light, all kinds of things that the epidermis protects the cells underneath from, including mechanical assault in the form of abrasion. Apoptosis is the name of the process by which cells can delete themselves. There are instances during development and under disease and stress conditions in which cells will deem a future looking very dismal for them, and they will just orchestrate a program of self-destruction called apoptosis. The dead cells that slough off the surface, the differentiation program of the epidermis, while being very specific to the epidermis, is considered by many to be a sort of specialized version of apoptosis. So the cells are working their way towards death and dissociation from the body. So apo is the prefix, tosis, the silent P is the root word, so apoptosis, programmed cell death. Here's a micrograph actually showing all the layers we just talked about. Here you can see the basal layer, and there's some melanocytes on the right. There's a cartoon drawn directly from that micrograph, so we can see a melanocyte over here. Too bad they didn't draw it brown, be a little bit more true to life. And you can see its long processes by which it will ship out vesicles full of melanin to all the keratinocytes around it. Here in the basal layer we can see a Merkel cell, a tactile cell, which in conjunction with a specialized dendrite of a neuron produces a sensitive touch receptor. So amazingly, through all those layers of the epidermis, if you touch the surface, it will deform the entire epithelium, and the cell will record those happenings and send impulses to the brain. Here's a Langerhans cell, a dendritic cell somehow able to move around within all these desmosome-linked keratinocytes, and is present as a first responder in case there's injury to the epithelium and some bacteria from the surface come right down in here and threaten the body with colonization. The spinous layer, you can see all the little dots represent the prickly edges of the cells where the really thick desmosomes have begun to form. The granular layer, you can see in this micrograph, are all these little purple dots you can see in the cells, which are the vesicles, the keratohyalin granules, and the lamellar granules. And then finally those cells die and produce all the layers of the stratum corneum, the cornified layers or keratinized layers. This would be essentially a picture of thick skin, because in a thin skin area, say such as on the inside of your forearm, there would only be a few layers of keratinized cells on the surface. So in part 2, we'll go on to examine in some detail the dermis, and then from there, in subsequent parts, we'll take a look at the appendages of the skin.